All right, so going into a little bit more detail for our neutrophils. So remember these guys with the granules, right? Fills, they're filled with granules. But for these guys, the granules stain relatively neutral, so a lot of times they're difficult to see in the cytoplasm. But these cells have that multi-lobe nucleus. So for these guys, their granules, as I just said, stain poorly. They contain enzymes and antimicrobial substances. So these are basically lysosomes, right? But also other stuff. So other names that you'll sometimes see for these guys too are polymorphonuclear, and again this relates to the fact that their nucleus tends to get elongated and what they call lobed. So sometimes that's abbreviated PMN, or sometimes just for short, polys, meaning again many, referring to the many lobes to their nucleus. Now sometimes two neutrophils, remember I mentioned last time a differential count that a doctor may order on your white blood cells, and this is where they're looking for the different types of white blood cells. Neutrophils are sometimes divided into the mature neutrophil that we're talking about here and the immature neutrophils, which their nucleus will tend not to have so many lobes. Instead, it will tend to kind of be like a horseshoe shaped. So because of that, a lot of times they're referred to as a band, B-A-N-D, bands. And those are the immature. So that means that, you know, they just came right out of the bone marrow. So if you're a really active infection where lots of neutrophils are being called up, then you'll see a lot of immature neutrophils in the blood. And of course, as they mature, their nucleus becomes more morphed. It becomes more lobed, right? Like you see here, kind of a zigzag. These guys are the most numerous, and of course their numbers increase. They're called up when we're dealing with infection, especially bacterial infection. So there's a saying that a lot of people learn in anatomy and physiology too, to help learn the normal distribution of the main white blood cells found in your blood. Right, so if you were to do a differential. Anyone remember that saying? Yeah, never let monkeys eat bananas. Right. So never stands for who? Neutrophils. Let, anyone know? Lymphocytes. Monkeys, M. Monocytes. Yeah, eat. Eosinophils. Bananas. Basophils. Right? So, and then remember, I'm going to talk about this in a moment. What do your monocytes turn into when they go into your tissues? They turn into macrophages, and they could also turn into, starts with a D, dendritic cells. Dendritic cells. Right. So yeah, these guys are normally the most abundant in your blood because they're there in case they're needed, so they can respond lightning fast. Right, so when we've got inflammation, they're called to that site. So they run right out of the blood, those large numbers, right into the tissue. They have the greatest killing power. Right? They're chock full of really damaging enzymes, antimicrobial substances. These are your superpower killers. With that, though, comes a very short lifespan, just a few days. They can release the contents of their granules, right, to the outside. So my nickname for these guys when they do that, I call them suicide bombers. Because they basically kill themselves in an effort to kill the microbes that are present in the area. So not only releasing their little bombs, right, they can also do what? And it'll kill them as well. Yeah, they can do phagocytosis, but what will they, that's not going to kill them. The DNA nets, right? When they literally shoot their DNA out of their cells, right? That's going to kill them. And that's going to help trap the invaders, though, right? So they're going to trap, some of them are going to set up the trap, others are going to release the bombs. Under certain circumstances. I have no idea. He asked, is that why they have the multi-lobes, so that they can do the net? I do not know how it is that that comes about, that they end up being multi-lobed. But it probably has to do with something like what's going on structurally inside those cells. But that probably makes sense, too, on why, like, when these guys use their granules, they don't regenerate them. And it's probably because, the deformity that happens to the nucleus makes it inaccessible, right? That information that they need to make more granules is inaccessible, I would think. And, I mean, really, I'm just assuming. But it makes me wonder, like, how is the nuclear envelope, the membranes that surround the DNA? I mean, like, how is this morphing happening? That's really a really cool graduate level question, I'm sure. Someone may have even investigated it. We could look into it. Not a high on my priority list, though. But definitely curious, right? Definitely makes us curious. So here's our big eaters, though, right? Macro, meaning big. Macrophages. So big eaters, big mouth, right? He's a big eater. So these guys differentiate, as we said, from monocytes. So we call them monocytes in the blood. And again, partly due to the fact that their nucleus stays intact, unlike the neutrophils. Although sometimes it tends to have a little indentation to it, almost kidney bean-like shape, or horseshoe-shaped. It doesn't reach the level of change that neutrophils do. They're sentinel. What does that mean? What does sentinel mean? They don't have to be called. They're already there, right? They're present in the tissues, and they're constantly sensing and surveying the environment, right? These are your watchdogs always on call. This is your alarm system always present. So, large numbers in your liver, your spleen, your lymph nodes, your lungs, and your peritoneal, your abdominal cavity. As we'll talk about later, your spleen filters what? It's actually blood. Blood. It's the one organ you can live without, although I'm sure you wouldn't choose to live without it. But if you have damage, right, something happens to it and they need to remove it, they won't replace it. No one gets a spleen transplant. Because you don't have to have it to survive. But obviously it's better to have it because it filters your blood. What some of the job of dealing with worn out, tired, debilitated red blood cells gets taken over by your liver if you lose your spleen. But that's not set up as its main job, right? So it's not as good at it as the spleen is. So it puts you at a little bit of a disadvantage, but you can still live. Your lymph nodes we'll talk about more when we get to adaptive Your lungs, right, you breathe in tons of crap, literally, every second of every day. So you got to have these guys, plenty of them on site to gobble up any of the stuff that gets in. And then your intestines, believe it or not, is a pretty big area that is susceptible to possible entry into the body. So that peritoneal cavity that, encases all the intestines, your digestive system, has to have a lot of protection. These guys are much more longer lived than neutrophils. They do regenerate their lysosomes. And so some students sometimes get confused too, because we were talking about neutrophils, right? And their granules, which some of them are lysosomes. Why is it when we stain monocytes under the microscope do we not see their lysosomes? Different substances, right? Because remember, between all the granulocytes, their granules stain different colors because of what substances are inside those granules. What dyes interact with those granules. So these guys have got, quote unquote granules, but really just lysosomes. So it's different substances. That's why we see this difference. And why you don't see their lysosomes. They don't get stained. That's the only difference. They just don't get stained. Because they contain a little bit different stuff than what neutrophils, and obviously different from eosinophils and basophils. They can be activated by other cells through chemical messaging, right? And the chemical messages unique to the immune system are referred to as cytokines, just like the chemical messages of the endocrine system are known as hormones, right? For the immune system, they're referred to as cytokines. And then other chemical signals as well. All of these can help make a macrophage a much more powerful killer. It gives it additional information. And so therefore it may use something else in its arsenal of attack. Right? In the DNA is encoded all these different enzymes they could potentially make, right, and different abilities. And sometimes, they need that little extra help that says, hey, you might want to go to the supersized bombs because the other ones are not working on this guy. So macrophages, there's some new designations out there. M1s are killer macrophages, right? So they're in that mode of killing, right? They're making enzymes to destroy, digest, kill pathogens. Some of them are in the healing mode. Now these are not set. A macrophage can switch between the different modes, right? It can be a killer 1 minute, and then later on, as the situation is changing, it may need to become a healer macrophage, right? Where it's cleaning up the mess and trying to heal up the tissue that's been damaged by the invasion. So you'll remember that we saw these in our granulomas, in that granuloma picture that I showed you from tuberculosis. And they also, remember what happens, how do we form a giant cell? Yeah, it's macrophages fusing together, or macrophages fusing together. And this usually happens within even a granuloma formulation, but granulomas are more than just fused macrophages. They're got T-cell involvement in that big grouping of cells as well. So I'll rewind back. Oh, I didn't put it in this PowerPoint. It's in the other one. Here, we'll open it real quick. I should have repeated that picture because it's really good. Yeah, this one. Right, so now look, oh, M1 means something to you now, right? It's a killer macrophages. M2 are healer macrophages, right? So notice that, you know, in this environment, they're more killers, right? They're trying to kill this bacteria, okay? Where down here, right, one killer there, mostly healers, right? They're trying to keep the surrounding tissue good. The problem is, right, they're not being successful at eliminating the invader. They're just fusing together. Foam cells. Anyone know what those are? Those are macrophages that ingest a lot of fat. And so they look foamy, frothy, because of all the fat, all the lipids that they ingest. So dendritic cells, I stole the cartoon from the website I sent you guys to. One of their really important tasks, although macrophages can do this too, the dendritic cells really do it for the most part. Just like a macrophage, they will take in substances, right? Foreign substances, bacteria, viruses, protozoa. They're going to take them in and they're going to ingest them. But they're going to go one step further. They're going to take that information and they're going to present it on their surface. And they're going to communicate with helper T cells to, you know, help out with this situation, right? With this invader we're dealing with. And so macrophages do this too. This is how part of the process of how they become activated into more powerful killers too, is if a T cell and a macrophage interact with each other. That's going to help activate that macrophage. In this case, the dendritic cell is activating the T cell, the helper T cell. So what does activate mean to you? It's turning on something, right? So they're recognizing each other. They're recognizing this pathogen. And remember what we said about T cells. This recognition is specific. It's the molecular level here, y'all. Right? Right down to little amino acids. They're recognizing this specific foreign invader. And so we're going to want more of these T cells that specifically recognize that specific invader. So one of the most common things that happens for lymphocytes when they become activated is they proliferate, right? They make tons of copies of themselves. So now we don't just have one cop on the beat that recognizes. We've disseminated. We've got a whole bunch of them that recognize the bad guy. I have no idea where my phone is. It's all right. Hopefully it won't ring. It's just dinging. Make sense? So we're going to go into, right, maybe unfortunately for you guys, I don't know. We're going to go in an adaptive, because T cells, right, are part of our adaptive immune response, this specific response that we can build up over time as we get exposed to different pathogens. We're going to go over in our adaptive chapter, and we're going to really go into the specifics of this interaction. So remember, we really want to focus in on the function of these cells. So for granulocytes, we've got those neutrophils, eosinophils, and basophils. And in general, right, their main job is phagocytosis and destruction of pathogens. Where eosinophils and basophils, right, little different action for these guys. They're involved in inflammatory responses and allergic responses, and you see eosinophils more so with parasites, especially parasitic worm infections. And then our other phagocytes, right, the monocytes in the blood that differentiate into macrophages or dendritic cells that stay in our tissues. These guys phagocytize, but again, the main job for that dendritic cell is presenting that information it's gathered to our lymphocytes. And then for our lymphocytes, right, they're part of our adaptive immune response, so we'll really get into their functionality a little bit later. They're only showing 2 cells there, and truly under the microscope, right, when you're trying to identify white blood cells, we just call them all lymphocytes because you can't tell the 3 different types from each other using that particular stain that they use for a differential. Okay, so, but what are the 3 lymphocytes? B cells, T cells, and natural killers. Who has specificity for antigens? T cells and B cells, right? Natural killer cells can't specifically recognize a specific pathogen on their own. They don't have those types of receptors, where T cells and B cells do, right? And as I said, we'll get into that in more detail in our adaptive chapter. So one of the things you've kind of already seen in that last picture of antigen presentation is that we need to have receptors on the surface of cells in order for them to be able to communicate with each other. They're going to send messages to each other as well in forms of chemicals, and for the immune system, those are cytokines, right? They'll actually be released by one cell, bind to a receptor on another cell, and it tells that cell to do something, just like hormones do. Except this is immune system, so they're cytokines. And then we've already seen adhesion molecules in action, right? They're going to help our cells grab on and travel into the tissue out of the bloodstream during inflammation. So sometimes those surface receptors are equated to being that cell's eyes and ears, right? It allows them to sense their environment, to see what's out there, to hear what's out there. The messages, as we said, are those cytokines. Those are the voices that they're calling out with, that someone else is going to hear and respond to. The adhesion molecules, as we saw, literally help them grab on, right? So they're very much like our hands. So the surface receptors, those are those membrane proteins, right? That's the job of some of the proteins embedded in the membrane of your cells. Receptors are specific to which molecules they bind. So most of us are familiar with this too as it relates to endocrine system and hormones. Things like insulin, right? Insulin only binds to the insulin receptors on the surface of the cells. And the cells are going to respond by saying, oh, thank you for letting me know glucose is on board. I will take some, thank you very much. Right? So same thing here, right? It's telling them a particular thing is going on, and they need them to do a specific response that that cell has the ability to do. And even like in our body with hormones, the receptor may be there, the hormone binds, and depending on what that cell is and what that cell is going to do, it may respond differently to that interaction than some other cell that has a receptor for that same hormone. I can't think of a specific example right now, but I know it exists. Too far removed from anatomy and physiology. But so, you know, of course we have terminology, right? The molecules that actually bind to a specific receptor is referred to as a ligand. And again, this is a signal we're sending. This is a message. So when that receptor binds to its specific molecule, its ligand, it's going to send a signal into that cell. And that cell is going to respond in a pre-programmed way to that message. It's going to do something. right? And that pre-programmed information is in its nucleus. So typically that message gets sent all the way to the nucleus, right? And that cell is going to probably make some protein, may even make another signal to send to another cell. So as I said, cytokines, right? This is a general name that we give to the molecules that are used by immune cells or cells to communicate with the immune system. There are numerous classes, right? So scientists like to group stuff, right? So, of course, we based on their general functionality. So there's a whole bunch of cytokines that are classified as chemokines because they're all involved in chemotaxis, right? Calling cells to the area chemically, right? They're sending this chemical message. The cells are moving in response to that chemical message. We've already talked about colony stimulating factors, right? These are going to be released by the body. They're going to stimulate the cells in the bone marrow to determine which populations of white blood cells we need at any one time. All right, so do we need a whole bunch of neutrophils to just keep coming? Then that particular colony stimulating factor is going to be sent that tells the bone marrow, neutrophils, please, right? Or macrophages, right? Or basophils or eosinophils or whatever it is that we need. Interleukins, inter means in between, right? Leukin refers to white blood cells, right? Leukocytes. So these are produced by white blood cells and typically to talk to each other. So there's a lot of them. They're commonly abbreviated IL for interleukin. And then, of course, they're numbered. And I forget how high the numbers go now. There's a whole book on just cytokines for the immune system. You thought endocrine was bad with hormones? Oh no. Immunology's got that stuff beat, hands down. There's a whole lot of talking going on in the immune system. They're noisy buggers. So tumor necrosis factor, as the name kind of implies, right, we discovered that these particular signals will lead to the killing of tumor cells. So these are really instrumental a lot of times too in initiating an inflammatory response to those type of cells being present. So this table actually gives you some examples. And again, these are just examples. By no means do I expect you to memorize them. I just want you guys to be familiar with the general classes of cytokines. You don't have to know specific ones. And so, of course, they're still actually very general for the first two. But for interferons, which we didn't even mention on the last one because we're going to talk about interferon in a separate section, these are involved in antiviral effects. So we'll come back to these guys. But you can see that there's three main interferons, and different cells are going to release those. What I want to look at right here is the interleukins, as I said. So they're just listing a few of them here, and as I said, they're commonly abbreviated IL and then a number. So you can see that the first one that we discovered is released by your macrophages and your epithelial cells. And it's going to help activate T cells. It's going to help activate macrophages. And it can induce fever. This is my favorite one, IL-2, released by T cells to cause T cells to proliferate. This one will literally be released by a cell and then bond to that same cell to get that cell to proliferate. It's like they're screaming at themselves like me this morning, you know. Don't forget your rain jacket, it may rain. Right? I was like, what? My husband's like, what? Nope, just need my rain jacket. No worries. It's like the brain won't register it unless you say it out loud. So for them, unless they scream out, I need to proliferate to themselves, they don't proliferate. But there are other actions that go on. So as I said, there's a huge list, right, of all of these. By no means, right, we're not a graduate level immunology class. We're not memorizing these things, right? I don't even remember half of the ones I memorized when I was in grad school. So you don't use it, you lose it, right? So, but, you know, the general understanding is what we're looking for here, right? We good? All right. Moving on to our adhesion molecules, right? So as we've said, we've already seen these guys in action, right? They are really instrumental in literally helping those cells grab on, move from the bloodstream into the tissue during inflammation. So they're on those epithelial cells, again, when those cells have been triggered to do such, right? Those adhesion molecules aren't always there because otherwise white blood cells would be grabbing onto them. And we don't need them grabbing on all the time. We need them grabbing on when we need them to come to that area. So it's going to help move them to that area. So for the next things that we're going to look at, and we're going to look at Tolllike receptor engagement. And so, obviously, as the name implies, Tolllike, so there are these sensors that they refer to as toll receptors. And then we found out about these other receptors, and they act very similar to those previous receptors. So they said, oh, well, we'll just say they're Tolllike, right? They're not toll receptors, but they're like toll receptors. What's the important thing here is what happens when these receptors are engaged. What's the outcome, right? What's going to happen when complement system is activated? What's the outcome of that? Right, how are we going to respond to these systems being triggered? What do we do when we detect long double-stranded RNA? Ooh, should be red flag, right? Double-stranded RNA. Do we have double-stranded RNA? No. That's foreign, right? What type of pathogen out there might be having that? Nope. Viruses. That's unique to viruses. And not all viruses, right? Just some, right? Because others are DNA viruses, some are single-stranded DNA or double-stranded DNA like us. But we certainly don't have double-stranded RNA. That is a dead giveaway. There's some type of invasion. And who has that are some viruses. So we have systems within the blood that can detect these signals of tissue damage or microbial invasion. For our innate immune system, what we're born with, right? This pre-programmed ability to know things that are not us is a very generalized system. So again, that double-stranded RNA is generalized, right? We don't have it. It definitely isn't us. But it's not specific. It isn't specific to a particular virus, right? It just says bad guy. Doesn't say who the bad guy is. Just says bad guy. So we're looking at patterns, right? Like double-stranded RNA that are not us. So what are some molecules that we've learned about that are associated with bacteria that we definitely don't have? That only they have? That's viruses. Bacteria, I said. He's stuck on that. Peptidoglycan, good, right? Their cell walls are made of peptidoglycan. We don't even have cell walls. We certainly don't have peptidoglycan. What about those gram-negatives? Remember, they have that outer membrane, but that substance that makes out the outer leaflet is very unique to gram-negatives. LPS, right? What does it stand for, though, y'all? Lipopolysaccharide, right? That's unique to bacteria. If they're motile, they're probably motile by what? Flagella. That protein associated with their flagella is unique to them as well, right? So those are very common, right? Pretty much all gram-positive bacteria, of course, are going to have lots of peptidoglycan. Gram-negatives are going to have lots of lipopolysaccharide. Some of those motile guys are going to have that flagellin protein. That's on the outside. These are all things on the outside of that bacterial cell even, right? That would help us detect their presence. So if we had general receptors, right, that recognize these general patterns, what peptidoglycan looks like, what LPS looks like, what flagellin protein looks like, that would be beneficial to us, right? So obviously having those receptors over time, we've kept them. Evolutionarily wise, the pressure has definitely been to keep those because they're protective for us, right? So respond to patterns associated with danger, right? We want to respond to those patterns. So, oh yeah, okay, there's peptidoglycan. No problem. Just sit back and chill. Not hardly, right? Peptidoglycan says danger to our body, right? Respond. Kill that invader, right? We want to directly be able to recognize and destroy that invading microbe. And we want to recruit other hosts' defenses. How do we do that? How do we get stuff to the spot? Yeah, cytokines are going to call it there. What's the general process of getting white blood cells to that area? Increased blood flow, and we get swelling. What do we call that response? Inflammatory response. Okay, so they like to use acronyms a lot in immunology, which I don't know how I feel about that because it gets hard to remember, right? The military is just as bad. When my ex-husband came back from boot camp, every other word was an acronym, and I was like, stop. Okay, what does that one stand for? What does that one stand for? Right? So we do this a lot in immunology too, right? We use a lot of acronyms. And then we get to the point where like, oh my God, we can't even remember what they stand for. Like CD, right? CD markers on lymphocytes. I had to look it back up because I was like, I've been using that acronym for so long, I forgot what it stood for. Clusters of differentiation, right? So for these acronyms, you know, we put them there because they're used, but I would caution you guys not, for some of them, don't use the acronym because you're not going to remember what it stands for, right? DNA. Sometimes you have to like think for a minute, don't you? Right? We use that acronym so often, right? Students always say to me, DNA. I'm like, okay, do you remember what that stands for? Because that's important, right? What does it stand for, y'all? Deoxyribonucleic acid, right? Deoxy is important, right? The sugar is missing an oxygen that ribose normally has, that sugar. And that's one of the ways we are able to detect DNA versus RNA. So those little details sometimes are important. So again, what we're looking for here is patterns, right? Molecular patterns, right down to the molecular level. And definitely we want to recognize ones that are pathogenic associated molecular patterns, right? The patterns that the bad guys have. So as you guys have already pointed out, cell wall components, things on the outside of the bacteria are very beneficial for us, right? So yeah, unfortunately for you guys, but you already knew them. You just named them all, right? We only missed one, right? We got our lipopolysaccharide, our LPS, right? We've got our peptidoglycan, lipotecoic acid. Do you guys remember who has this? Gram positives or gram negatives? Gram-positives, right? It's kind of the rebar, helps hold together the peptidoglycan layers because there tends to be a lot of them. Lipoproteins are also very commonly associated with bacteria. Flagellin subunits of the flagella. And then viral RNAs. Again, their RNA is very different from ours. We modify and modify our RNA in a particular way. So if RNA comes in that doesn't look like ours, we're actually able to detect that. And especially if it's double-stranded, right? Because we never do that to our RNA. So again, there's lots of different acronyms out there, right? But the key words that we're keying in on here is that we're looking at patterns, right? At the molecular level that are associated with pathogens, with microbes, with danger, right? All these guys are bad news. But truly down at the molecular level, we're looking at components associated with the cell wall and other components of pathogens, of the bad guys. So let's see if I can get this to work today. So here we have these receptors, right, in the cell membrane. Let's plug it in.
A number of different components.
All right, now I got the volume. Let's play.
Structures on the surfaces of phagocytes, called pattern recognition receptors, make it possible for the phagocyte to immediately recognize a number of different components of infectious agents, referred to as pathogen-associated microbial patterns. Toll-like receptors are a class of pattern recognition receptors that play a critical role in the early innate immune response to invading pathogens. Several different toll-like receptors located in the cell membrane or endosomal membranes of phagocytic cells have been identified. They recognize lipoproteins and lipoteichoic acids, viral double-stranded RNA, lipopolysaccharide, and bacterial flagellin.
All right, so notice on the surface of the cell, this one, Toll-like receptor 3, what are they saying it's responding to? Viral double-stranded RNA. So it makes sense that this receptor needs to be where? On the inside of the cell. Because only when the virus gets inside of the cell is then the double-stranded RNA going to be accessible for this receptor. If we had that receptor on the surface of our cells, it would do us no good, right? Because the virus has what around its DNA or RNA? Capsids. And what are those capsids made out of? Protein. Right? We're not going to recognize it. Make sense? Good observation. All right, continuing on.
Some toll-like receptors exist as monomers, whereas others exist as dimers. When a toll-like receptor interacts with a particular pathogen component, it activates specific cytoplasmic adapter proteins such as MYD88, MAL, TRIF, and TRAM. This activation initiates a signaling pathway to defend against a specific type of pathogen. Lipopolysaccharide is a pathogen-associated structure produced by gram-negative bacteria. It first binds to lipopolysaccharide binding protein and then associates with a cell surface protein CD14. The CD14 with bound lipopolysaccharide binding protein then activates TLR4. The activated TLR4 then interacts with and activates specific cytoplasmic proteins. The interaction of TLR4 with these cytoplasmic proteins activates other proteins, allowing them to convert inactive transcriptional activators such as NF-kappa B into active forms. The The activated NF-kappa B moves into the nucleus and binds to specific sites on DNA. Binding of NF-kappa B to specific sites on DNA induces transcription of genes for cytokines, including interleukins and tumor necrosis factor. These cytokines move out of the phagocyte and stimulate various aspects of both specific and nonspecific immunity.
Who's asleep? Come on, come on. It gets really boring there in the middle, right? Even I zone out, right? Again, all those acronyms, right? We should have named things like, you know, the George protein or something like that. I don't know. I get lost, right? But it's okay, because do we need to know all these details? God, no, right? Okay. But what's the overall story here? Okay? So, in general terms, what happened here? So we have receptors that are recognizing a particular pattern, right? A particular molecule we know is associated with the bad guy, lipopolysaccharide. That receptor is triggered. What happened in general terms? What happened once this got triggered? A whole bunch of stuff happened in here, right? But it's basically passing a signal, right? Where did this signal go? To the nucleus, right? And what happened there? It activated something, right? We made something. We sent out a signal, probably messenger RNA, to make other molecules that were then released by that cell. So that cell received a message, and he even passed that message on to other cells. And he himself, depending on the cell, maybe even had responded to that in a particular way, in addition to what they're showing here. So they didn't even really get very specific, right? They said, you know, these were the possible cytokines that could have been released. I got news for you. For certain situations like this, we know exactly what happens, down to this level of molecular interaction. So, take-home messages: message received, message sent on, right? So Toll-like receptors are anchored in the membrane of sentinel cells, right? Our macrophages, our dendritic cells, cells lining sterile body sites, right? Where no bad guys are allowed to come in, right? So if they get anywhere near, we're going to send off the alarm. So if bad guys come in, right? And then, of course, sometimes it's stuff that'll actually get into those cells, right? We need detection on the inside. So again, notice we can even detect things like bacterial DNA inside of our cells, double-stranded RNA and single-stranded RNA that would be viral in nature. Following detection, the signal is transmitted to the nucleus. Okay, so what kind of things do we want to happen after this signal goes into the nucleus? So it's going to release cytokines, right? But if we've got an invader, some of those cytokines need to tell the cells to do what? Come here and help me, right? And what's the general term we call for that? When we want cells to come to that area. We want... Remember, you can say it in one word that starts with an I. Inflammation. Remember I said we started out with inflammation for a reason because I was going to make you say it to me like a gazillion times? We want an inflammatory response. And definitely if it's a viral signal, we want an antiviral response. Which is a little bit different than how we respond to bacteria. But this is one of the important outcomes that is going to happen from Toll-like receptors being engaged.
So this is all happening before the inflammation of that?
Yeah, so she said this is all happening before inflammation. Yeah, this is the signal that causes inflammation to happen, right? So remember, when our cells get damaged, they're going to release things that are going to elicit the inflammatory response. But also, macrophages may be on scene, and they detect the LPS like we saw. And then they're also going to send out the signal for an inflammatory response. Or in the case of it's viral, then it's going to be a particular signal for a viral response, an antiviral. Yep. So, yeah. Our system is kind of, you can almost say, is kind of redundant too, right? So you don't just have the cells themselves that are damaged calling out. You've got these sentinel cells there, like the macrophages, the dendritic cells, and maybe other cells there that have specific receptors so that we can be more specific in our response even. You know, we're going to be like, oh, bacteria on board, right? Because I see lipopolysaccharide, right? We definitely need neutrophils, like all you can send them now, right? Or, ooh, it's a parasite. We need some eosinophils on scene, right? So yeah, the signals get very specific depending on what receptors get triggered. But again, inflammatory response is gonna get the cells there, right? That general response of the epithelial cells expressing those adhesion molecules, loosening up those junctions between the cells, allowing the white blood cells to come in between and that fluid to also leak into the area. So NOD-like receptors, they're found in the cytoplasm. So we've got receptors on the cell membrane. We've got ones within endosomes within the cell. Oh yeah, we've got it all covered though. Even within the cytoplasm, we have receptors. So if a bacteria gets in to the cytoplasm, we've got peptidoglycan detection there. We've got, single-stranded RNA that's uncapped because we cap ours. Flagellin if it gets in. Ones that will detect cell damage within the cell. So some of them, some NOD-like receptors, join cytoplasmic proteins to form an inflammasome. Well, that just tells you right there. What's the response going to be? Inflammatory response. Right? We're going to call all our friends to come help us. So, triggering of receptors is step 1. Sending out the message is step 2. And that message is going to be an inflammatory message, especially if it's bacterial. We got to get the white blood cells there to eat up and kill these guys. Right? If it's viral, it's going to be a little bit different. I don't know if we're going to get to viral today. Probably not. I think I'm going to end with the end of that PowerPoint, because I forgot to post the next one for you guys anyway, so it's all good. I will be more Johnny on the spot than I was with this last one. But please take a second and don't pack up. Those of you guys that were here on time know that I forgot to, I tried to take roll. It wasn't I forgot. I tried to take roll and it wasn't participating. So let's see if it will participate. And there is one thing in Canvas I want to show you guys before I end my recording. So please stop packing up. We have time. Packing is noisy. Okay? All right. All right, so fingers crossed. So you guys have a new assignment. Really, you have a new assignment. All right, it probably won't get recorded. I'm going to tell you about it. Actually, you can see it right here. I don't have to go to it, right? So it's called an agreement. You need Lockdown Browser to complete it. So you've either got to download it. I gave you guys a link already, right? Yeah. You got to download it, The link should be under the exams module, by the way, and the instructions. You either download it at home, or you can go upstairs to the second floor, open computer lab. You have to have your ID, by the way, to swipe in there. Or you can go to the Hibernia Center where we took our test, if some other class isn't already in there. There you don't need your ID, but you will log in like you did before with an access code. If you don't know it, they'll look it up for you. But you might, if you plan on using that one a lot, write it down or put it in your cell phone, like a lot of us do nowadays. They have the lockdown browser, so don't open any other program on the computer. You click lockdown browser, it takes you right to Canvas, you log in. Your test results have been available to you since Friday. I highly suggest that you look at them, especially if you haven't looked at them yet. And then once you get done looking at them, do this quick agreement assignment. It literally is like 5 or 6 questions, a whole bunch of yes or no's, and one very short answer where I ask you a question about whether you bought the book and why you bought it, why you chose what you chose. Okay, it's so simple, easy. Okay, but it has to be open in lockdown browser, and this is kind of my additional check that you've made the extra effort to review your exam. Also, the first question says, have you reviewed? And the instructions for this assignment say, review your test results before taking this assignment, right? So you will lose points unless you lie. The first question is, did you review your exam results? You want to click yes. That's the correct answer. Y'all understand this?
No.
Okay. All right. So do that by Tuesday, and then let me get on the other computer and take attendance. So here, I'll end the recording.